All right, last chunk of Lecture 6. And again, this stuff will be assessed on the quiz on Friday. I am assuming that you'll be using your notes and that sort of thing, so expect short answer questions versus only multiple choice so that you can phrase your answers the way you need to in your own words instead of just picking the right answer. So, this lecture is all about different processes the cell can do. We've talked about how energy is used in the cell. We know that's a characteristic of life, but we also need to get the materials in and out of the cell. So we're going to talk a minute, or probably more like 15 minutes, about how cells get molecules in and out. And of course, we need to go through the plasma membrane. And the function of the plasma membrane, which we saw in the last lecture, is it regulates movement in and out of the cell. That membrane, kind of like the surface area, when we talked about the surface area to volume ratio, is the gateway to get molecules that we need into the cell and waste products out of the cell. So by nature, this plasma membrane is considered selectively permeable. So selectively means there's a selection process of how if molecules can come in and out, and it's not permanent. So permeable is a similar word to permanent. So its permanent nature is selective. Some things can never get in or out of the cell. Other things can freely. And a third option is that molecules can get in and out with some help. So some pass freely, some get transported, and then some are prohibited. And it's based on size, it's based on charge, it's based on a few factors. And I'm not going to go into all those factors, but I do want you to know that there's 3 main ways to enter the cell: passive transport, active transport, and bulk transport. So I've got a couple slides on each of these, plus some pictures that we'll look at. Okay? And so again, I want you to remember that it's because of the components of the membrane, like the phospholipid bilayer and the proteins that are embedded in the plasma membrane, that enable this property of being selectively permeable. Okay, so passive transport's the first one. And the reason we call it passive is because there's no energy required. This is just simple diffusion, molecules moving down their concentration gradient until they equal out on both sides of the membrane. So they move from areas of high concentration, where there's lots of them, and they spread out to areas of low concentration. So the cell does not expend any additional energy. The molecules are already moving around, so it uses that natural motion of molecules Molecules are just going to do this no matter what. Even if there's not a cell membrane around, molecules are going to spread out naturally. And therefore, some of the molecules will just slip between the phospholipid molecules. We said in that fluid mosaic model that it's not, they're not all bonded together, that those little phospholipids can kind of move around and are fluid. So this has nothing to do with a cell. This is just dye in water, right? So if we drop a crystal of dye into water like a fish tank, over time it's going to dissolve. But not just that, but the dissolving is going to become uniform throughout the whole fishbowl. So if you think about the pigment molecules in that crystal, when it's solid, all of the pigment molecules are concentrated together. This is an area of high concentration of dye. And then it starts spreading out, and the water starts spreading in towards that. And after a while, it may take some time, there should be an equal distribution of molecules, because those dye molecules are moving from their high concentration into areas of low, and then the water is also moving from areas of high concentration to low. Now there's some specific types of diffusion. Again, diffusion is just the idea that molecules move from high concentration to low concentration. If it's diffusion across a membrane through a protein, We call that facilitated diffusion. And you can go back to the last lecture and look at those cell pictures, and you'll see some more as well. And we already saw one earlier in this lecture, where there's those transport proteins that move molecules, and some of them work by allowing molecules on the high concentration side to move through the membrane to the low concentration side until they reach equilibrium. Osmosis, and you may have heard of the movie Osmosis Jones, which doesn't have much to do with osmosis, truthfully. Osmosis is a specific type of diffusion when it happens with water. Water is super important. I think we've talked about water in maybe every single lecture so far. Water is very important to life, and so how it moves in and out of cells is also very important. And it moves from areas of high concentration to low concentration when left on its own. And that will happen across the plasma membrane. So here's a picture of facilitated diffusion. So let's imagine there's these big green molecules. We call them solutes. It's nothing specific. They're way too big to be going straight through the plasma membrane. We see our little phospholipids here with their fatty acid tails. However, when they come in contact with this carrier protein, the carrier protein will allow them to move through the membrane. And so over time, the outside world and the inside world of the cell should become equal when it comes to this particular green molecule. With osmosis, sometimes this is going to occur inside cells when the solute is too big to move through the membrane. Okay, so what we're imagining is these green dots are just too big to go across the membrane. So in order to change the concentration, while it would be great if these green molecules could just go down and even out, because there's a lot more above this red membrane than there is below, but let's say the solute's too big. It can't get through. Well, in order to balance it out, water, which is very small, H2O, can go through most membranes, water will enter. And so what this does is it spreads out the green molecules until they're the same concentration. They'll never be the same number of green molecules inside and outside, but they can be the same density or thickness or crowdedness, however you want to think about it. The concentration is the same. So overall, we can actually see this in this type of reaction. So if you imagine this is a type of flask, a volumetric flask here, that you can see that when there's a 10% solution inside and a 5% solution outside, that's how much green molecules there are. But if you think about the water, there's less water inside than outside. So over time, water will enter, and sure enough, we'd be able to see this line of liquid increase. Do you notice anything about the intensity of green over time? Well, sure, it's dark over here and it's lighter over here because water has entered and has spread out those green solute molecules. So what does this mean in real cells? Well, we need the inside environment and the outside of the environment of the cell to be equal. Equal in solutes, equal in water. If not, molecules will start moving in and out of the cell, and it'll disrupt homeostasis. It won't keep the cells inside those stable internal environment conditions that we need for life. So when the inside and outside world is similar, we call it an isotonic solution. Iso means equal. So that means there's no net gain or loss of water. Sure, water is moving in and out of the cell, but it's moving in and out at an equal rate. So this makes cells look like nice normal cells. It means that red blood cells are nice and circular with a little dimple in each side. It means that plant cells fill up to the nice cell wall. It has this nice central vacuole that's nice and full. So in our world, our isotonic solution is 0.9% saline solution, which is about the same as contact solution. It's the same that if you get dehydrated and have to go to the hospital, the IV they put in you is not just water, but it's saline solution. Hypotonic solution is when the concentration of water outside the cell is greater. Okay, and so this is going to make water rush in, and the cells will swell, Some cells can actually rupture. In plant cells, they don't really rupture because they have that thick cell wall around them, but they do become very, very full, and it can put pressure on the internal organelles of those cells. Okay, so hypotonic solution means the cells get big like a hippo, and they're getting big like a hippo because there's a higher concentration of water outside the cell than inside. Okay, hypertonic is the opposite. So in the medical field, anytime you see hypo, it means low. Anytime you see hyper, it's high. And that's actually talking about the solute, not the water. Okay? So in hypertonic solution, this is when your cells get dehydrated. That there's less water outside the cell than inside. And so water from the cell is lost to the environment. It leaves the cell, leaving the cell shriveled, wilting your plants, so that because those individual vacuoles and cells are no longer full of water, so they shrink away from the cell wall. And that's in hypertonic solution. So hypotonic, I think big like a hippo. Hippo, hypo. Okay, it's close. And hyper, I think about a skinny hyper kid getting and very shriveled up. Okay? It may not work for you, but it works for me. Active transport is when the cell still moves molecules across the cell membrane, but it takes energy. And if you notice, it's actually moving against the concentration gradient, meaning it's going from low concentration to high concentration. This requires a transport protein, but it also requires ATP. So this is one of those actions that the cell needs that, let's say, we don't want to reach equilibrium. We want to keep that gradient of high versus low. Well, it takes energy to do that. It's like walking uphill, going against the gradient. It takes energy to do that. Sledding down the hill doesn't take much. That's like passive transport. Okay? Walking up that hill is active transport, going against the gradient. The third type is bulk transport. This is a type of active transport, so it does require energy, but this is for macromolecules that are too large to go through just the individual transport proteins. And usually what happens is that cell membrane wraps itself around the molecules and pulls it in the form of a vesicle, because we already talked about this. The vesicle, this plasma membrane, all the internal membrane systems are the same phospholipid bilayer. So if we're bringing big molecules into the cell, we call it endocytosis. If we push them out of the cell, it's called exocytosis. So exo like exit, endo like bringing in. Here's some great pictures of how this actually works. So this is exocytosis. We have this vesicle. It's a double membrane phospholipid bilayer. We've got these green molecules we want to push out, and it fuses with the plasma membrane and pushes out the vesicles. And guess what? The same thing happens with endocytosis, only in reverse. So the cell membrane pinches in and forms its own vesicle to be brought in. This does require energy. It's not going to happen on its own. And usually it has like receptor proteins that start and stop this process. Here's actual pictures. So here's that receptor protein that says, oh, okay, we need to pinch this part of the membrane in, and then it forms its own internal vesicle, which is pretty cool. Okay, so in this chunk, I want you to be able to tell me the 3 divisions of cell transport, and I want you to know which ones require energy and which ones don't. And we're going to circle back and talk about the plasma membrane and what type of molecules are involved in each type. You can look at the pictures and see that as well. And then I need you to know what happens to a cell in a hypertonic and a hypotonic environment. Go back through the notes to make sure you know the answers to these questions. All right, coming up later this week will be Lecture 7. That should be uploaded by Wednesday.